Okay, this begins chapter 8, where we're going to talk about the metabolism of our microbes. And what is metabolism? It's all the chemical and physical workings of a cell. And we have two types of chemical reactions that happen. First is catabolism. This breaks down large molecules into small ones, where we break down proteins into the amino acids that make them up, or we break down the carbohydrates into the sugars. It's done to release energy. And then anabolism is the other chemical reaction where we're actually putting together the smaller molecules to make the larger macromolecules, where we put the amino acids together to form a protein, or we put the sugars together to make a carbohydrate. And this is going to require us to put some energy into the process, so it's going to require the input of energy. And this is just showing you here the way that we have our sources of energy here, for which is our carbohydrates, our lipids, and our proteins. And when we break them down, we're going to release the energy. And then here with anabolism, we got to have energy in order to break down the lipids and the proteins and the carbohydrates and the other macromolecules, or in order to put them together. Now, to review enzymes, remember that enzymes are just proteins, and we call them catalysts because they increase the rate of a chemical reaction without becoming part of the products or without being consumed in the reaction. So they just assist with and make the reaction happen easier. In this graphic that we have here, the reactant is the boulder. And the activation energy, the energy needed to move that boulder down that cliff. The enzyme is the crowbar. That's going to reduce the energy that we need. It's going to help us. So it's the enzyme. And the product, of course, is the boulder when it gets to the bottom of the cliff or the hill. Now, this is enzyme structure. And you remember that an enzyme is what a substrate will bind to. The enzyme will promote a reaction by serving as a physical site upon which the reactant molecules or the substrates can be positioned for various interactions. And here the metallic cofactor, that's a non-protein inorganic molecule. The coenzyme that you see here, these help enzymes do their job. They're organic molecules, and they work with these apoenzymes down here to help change the substrate. And what the apoenzyme is the active site where the substrate binds. And the coenzymes often contain vitamins. Now, if we classify enzymes according to characteristics like where they happen, what happens, and the substrate, this gives you some examples of some names of enzymes. Lactase, for instance. Naming enzymes involves two parts. There's a prefix or a stem word, and then the substrate with ASE attached to the end. So the substrate is lactose, so we put ASE on the end of it for lactase, and it's going to break down lactose. Penicillinase, the substrate is penicillin. For DNA nucleotides, the substrate, the common name of the enzyme is DNA polymerase. So this is just showing you how some of them are named by where they occur or the type of the action or the substrate. Now, energy is the ability to do work or to cause change. And we have different forms of energy. We have thermal energy, radiant energy, electrical, mechanical, chemical, atomic energy. Cells manage their energy in the form of chemical reactions that either make or break bonds and transfer electrons. When electrons are lost from a chemical substance, it's called oxidation. When electrons are gained from a chemical substance, it's called reduction. Redox reactions always happen in pairs. There's always an oxidizer that donates an electron and a reducer that accepts an electron. And those together, the oxidizer and the reducer, that's the redox pair. And this salvages the electrons and the hydrogen atoms with their energy. So the energy is released and then can be captured. Our first concept check in this chapter is if a molecule has been reduced during a reaction, it has gained electrons and hydrogens.